In Chapter 11, we're going to talk about the second type of cell division, meiosis, and meiosis is highly linked to the process of sexual reproduction, so we will also talk a little bit about that process. We will focus on it mainly from an animal standpoint, but we will talk a little bit about plant and fungi, sexual reproduction as well. So what is meiosis? Well, meiosis is a key type of cell division that's going to increase what we call the genetic variability. If we think about an asexual organism like a bacteria, it starts as one single cell and it divides into two genetically identical cells. That isn't necessarily a problem. Bacteria are very successful, but the only source of genetic variability is then mutation. Meiosis, on the other hand, feeds into the process of sexual reproduction, which is all there to increase genetic variability, allowing evolution to function. So meiosis is a special type of cell division, and it reduces the ploidy number by half. It also will allow some swapping of genetic material that is going to increase the variability. That's going to be the key to sexual reproduction. So we're going to focus heavily on the behavior of chromosomes during meiosis, where they move. There are two stages to meiosis, so we'll track them in both the first and second meiotic divisions. We're going to talk about the cellular events that take place in meiosis, and then look at the difference between meiosis and our previous division, mitosis. We'll then talk about the process by which it increases genetic variability, and talk a little bit about how the haploid gametes are produced. And down the line, in the next few chapters, we will see how this feeds into our concept of genetics and evolution. So let's get some definitions out of the way. Asexual reproduction. This is generation of genetically identical offspring. They're essentially clones. This is barring any sort of random mutation. But you could think about this like you take a tree and you cut a little bit off of it and you stick it in the ground. Sometimes it will grow roots and you grow a new tree. But that tree is genetically identical to the one that you cut it off from. So that would be asexual reproduction. Sexual reproduction, on the other hand, involves creating what we call haploid cells from the parents, which will then fuse to form a new diploid organism. This organism, it has the genetic information from half from one parental lineage and half from the other parental lineage, and it can create new combinations of genetic material in this way. Sexual reproduction has to involve meiosis because we need to reduce the ploidy, and then the process of fertilization where sperm and egg come together. So let's review ploidy. So here we have an organism that has three different types of chromosomes: a red chromosome, a purple chromosome, and a yellow chromosome. In a haploid cell, or 1N cell, there is one copy of each of these chromosomes per cell. In a diploid organism, there are two, hence 2N, two copies of each of these chromosomes. So 1, 2, 1, 2, and 1, 2. Now, each of these chromosomes is going to have come from one of the lineages, either the maternal lineage or the paternal lineage. I can't tell you which based off this diagram here, but that means that you could have slightly different versions of the genetic information on this chromosome compared to that one. That's interesting. That's genetic variability right there. So how does this all fit together? Well, we're going to focus a lot on meiosis, but let's review the sexual reproduction life cycle for humans here. So in humans, we have the maternal lineage that has ovary tissue and the paternal lineage that has testy tissue. The ovaries, the cells in there will go through meiosis and they will produce a haploid gamete called an egg. The testes tissue will do the same thing, but it will produce a haploid gamete called the sperm. Okay, those are 1N. They have one of each of the 23 different types of chromosomes that make up the human genome. Okay, and then we have fertilization. Fertilization is where these two haploid gametes fuse together. So the sperm fuses with the egg. I think you can see where this is going. 1N plus 1N makes 2N. We form a cell that is diploid, 2N, called the zygote. The zygote is the first diploid cell that will divide by mitosis to produce all of the somatic cells in the mature organism. But for humans, to produce the gamete cells, we have to talk about this special division, meiosis. As far as an overall goal, the purpose of meiosis is simply to reduce the ploidy by half. So if you are a diploid organism, it will produce cells that are then haploid. I think you can see based off the previous slide how that feeds into the sexual reproduction cycle. If we did not do that, say if we didn't reduce to haploid here, the egg would be 2N and the sperm would be 2N, and then we'd end up with a 4N zygote, and then we'd repeat the process, and then it would be 4 and 4, and then we have 8N, right? And we'd go on and on and on, and it would just run out of control. So the meiosis, reducing the ploidy by half, is a key step to sexual reproduction. Now, this ploidy reduction is going to occur because there are two stages to division for meiosis. We call them Meiosis I and Meiosis II. Before meiosis, there is interphase that consists of G1, the S phase where the genome is duplicated, and the G2 phase. All right, you might be wondering, why is the genome duplicated if we're trying to reduce the ploidy? We'll see that here in the next diagram. We have meiosis, our overview here. So in this cell, we have one type of chromosome. And here in the diploid cell, we have one copy of each of this chromosome. Here in the diploid cell, we have the one chromosome. We have the paternal version and the maternal version. So these are homologous chromosomes. All right, what's the first thing that's going to happen? Well, in S phase, these are going to get duplicated. Looks very similar to mitosis, but don't worry, it will get different. So these homologous chromosomes replicate. Then we go into the process of meiosis itself, and we'll focus on Meiosis I here. Now, something interesting happens in Meiosis I that is different from mitosis. Instead of all the chromosomes like lining up at metaphase, what they do is the pair of homologous chromosomes pair up. So we call these sister chromatids. The two blue ones are sister and the two purple ones are sister, but they're homologous because they're the same chromosome. These two chromosomes actually kind of butt up against each other and can swap pieces with one another. We call this process recombination or crossing over. We will come to talk more about this in the genetics chapter. This is a very important part of genetic variability. At the end of Meiosis I, these two sister chromatids will pull apart, and we will have our two cells here. Now you'll see it's calling them haploid. They kind of technically are haploid because they only have one version here. Don't worry about that too much. That's kind of a technicality here. What we see, though, is one has primarily the paternal and one has primarily the maternal. Then we go into Meiosis II. Here is where the ploidy is reduced. So these sister chromatids get pulled apart in Meiosis II, and we end up with 4 cells, each with one copy of this chromosome. You'll see one of them looks like the original paternal, one of them looks like the original maternal, and then we have these 2 recombinant versions in here with little bits that have swapped over. So this process differs because we end up with haploid cells at the end of the division. Let's look at some key pieces to this now in more detail. Okay, one of the key pieces in Meiosis I is Prophase I. So all the cell cycle stages are called the same thing. We just have step 1 when we do the first meiotic division, and Meiosis II, the second meiotic division. So in Prophase I, the homologous chromosomes are brought close together. So here you can see the red sisters and the blue sisters. They are brought into physical proximity. And we get this tight pairing called synapsis. This allows the swapping of pieces between the homologous chromosomes. We call this crossing over or recombination. Now, let's look at that closer. Okay, so let's do a hypothetical here. Here's our chromosome, and on our chromosome are genes A, B, and C. Okay. You can see on the blue version, we have big A, big B, big C. Those are certain what we call alleles, versions of a gene. On the red chromosome, we have little a, little b, little c. Same order, same genes, just slightly different versions of them. I'll show you some examples of alleles in a moment. All right, so during recombination, these two homologous chromosomes get close to one another and can swap pieces. So now on these new chromatids, we have big A, big B, little c versus little a, little b, big C. We've swapped. We've created what we call a new combination of alleles here. And this structure, just to be fully clear, gets called a tetrad sometimes. It has two non-recombinants on the outside and two recombinants on the inside. So this is creating new combinations of allele, like I said. So what is an allele? Why does this matter? Well, we'll look extensively at pea plants in genetics because one of our earliest models for genetics was the pea plant. But here's some examples of alleles. So it could be that, I don't know, seed shape. That's our gene A. Okay, big A leads to a round seed shape, where little a leads to a wrinkled seed shape. Or big B could be for seed color. Big B could be yellow. Little b could be green. Flower color, that could be gene C. Big C could be purple. Little c could be white. We could have something like pod shape or pod color or flower position, or is it tall or dwarf? Anyways, let's look at those first three together. So in this example, this chromatid here has all dominant alleles: seed shape, seed color, and flower color. If we look in the middle, though, we get a new combination of alleles here. We get big A, big B, but then little c. So instead of being a round, yellow, purple flower, we would see a round, yellow with white flower potentially coming from that gamete. That's a new combination. So each meiotic division is crossing over differently. It's creating new combinations. So that's in Prophase I. That's a really key piece to meiosis and its ability to create genetic variability. Metaphase I. When we get to Metaphase I, there are two possibilities for how homologous chromosomes could line up. So here we have a system where we have two types of chromosomes, a big chromosome and a little chromosome. You can see we have a, let's say, maternal version and a paternal version. So we need to look at this combination here. The combination here, we have the maternal big chromosome, the blue one here, and the maternal little chromosome here on the left. The two red ones are on the right. If we follow meiosis, these two will get pulled together, and these two will get pulled together, and will create blue big and little gametes and red big and little gametes. Let's look at the other possibility, though. When they line up at metaphase, they could be opposite. The blue one could be on the left for the big chromosome, and the red one could be on the left for the little chromosome. Now when we follow meiosis, you can see we get a different combination here. We get a blue big chromosome and a red little chromosome in these gametes, and we get the reciprocal over here. This is entirely random, and it has an equal probability of each of these things happening. But you can see that this gamete has different information than this gamete and has different information than this gamete. It's got different alleles in there. So we're providing different arrangements that provide different gamete combinations here. So this is an example of just two chromosomes. Remember, you have 23 different chromosomes. So I think the math is 2 to the 23rd power. So there's 23 different chromosomes and two possible ways they can line up if we do all those combinations. I think that's over 8 million combinations. So just right there in this process that we call independent assortment, we get a lot of genetic variability as well. not even to mention the crossing over or any sort of mutation that could happen in there. So we have two possible mechanisms for creating genetic variability in the gametes here. We have one, the random recombination of homologous chromosomes during prophase I, and we have the random lining up of homologous chromosomes at metaphase of the metaphase plate of metaphase I, called an independent assortment. Both of those things contribute to different combinations of alleles there. So once we go through those, then we do anaphase I and telophase I. In this example, these cells have three types of chromosomes, so you can see they're getting pulled apart. The two purple ones line up on top with one of the blue ones, and the reciprocal down here. So we go through anaphase, they pull apart. Sister chromatids are still attached. We divide the cell here in two, and here's where the kicker comes in. So that's meiosis I. So the big kicker here, though, is that going from meiosis I to meiosis II, there is no duplication of the chromosomes. This is going to lead to the reduction in ploidy. So let's go into the meiosis II. So we'll have prophase II, then we have metaphase II, and then we have anaphase II. This is really where the key difference occurs. Anaphase I versus anaphase II. So remember in anaphase I, the sister chromatids were still attached. So the homologous chromosomes pulled apart, but the two sisters were attached. In anaphase II, the two sisters get pulled apart, and this is going to lead to our 4 haploid gametes being produced at the end of this. So metaphase II occurs, and we pull apart, and then we end up with our haploid gametes down here, 1N. So let's look at the full picture here. So we started with 1 2N cell, and then we ended up with 4 1N cells at the end of this. This is very different from mitosis, right? Mitosis keeps the ploidy the same. Meiosis reduces the ploidy. So key points: prophase I, that crossing over; metaphase I, that independent assortment. Then we look at anaphase II. That's where the sister chromatids get pulled apart and we end up with the haploid gametes, because there was no duplication of the genetic material in between meiosis I and meiosis II. All right, so let's examine some of the differences between meiosis and mitosis. Well, right off the bat, right, meiosis has 2 steps, and the overall goal of meiosis is different. Meiosis is reducing the ploidy, whereas mitosis is keeping the ploidy the same. So we have 2 key big events in here: crossing over and independent assortment, that are very different in meiosis I. Meiosis II looks very similar to mitosis, but because we haven't duplicated the genetic material, we end up with haploid gametes in there. So you can review this if you need to see all the differences spelled out for you, but let's look at a review animation.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into 2 stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains 2 uncoiled, spread-out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with Prophase I, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in Metaphase I, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During Anaphase I, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters Telophase I with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, Meiosis II. However, unlike Meiosis I, DNA does not replicate before Meiosis II begins. Once again, in Prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During Metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During Anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during Telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with four genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has two stages of cell division, called Meiosis I and Meiosis II. During Meiosis I, homologous chromosomes separate to produce two haploid cells, each containing chromosomes in the form of paired sister chromatids. In Meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces four genetically different haploid gametes. And here are some key points to remember about Prophase I. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of four chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Let's go ahead and do a review question here. Let's think about what would happen if animal gametes were made by mitosis instead of meiosis. Go ahead and pause the video to think about that one. All right, so let's think about the difference between these two. What are the end results? Well, meiosis results in 4 haploid gametes at the end of the division, whereas mitosis results in 2 diploid cells. If you started with a diploid, the ploidy stays the same. So if you made your gametes by mitosis, you would end up with diploid gametes. Those diploid gametes would fuse, and then you'd have a 4N cell, right? And then if that 4N cell made its gametes by mitosis, you'd have 4N gametes, and those two would fuse, and you'd have 8, right? And then the process would continue, and after a couple generations, you'd have more chromosomes than you know what to do with, right? And they'd just fill up the cell. So meiosis is really critical for keeping the chromosome number the same. But also, let's think about genetic variability. You would have no crossing over, no independent assortment, so you would not get new combinations of alleles being produced in there. You'd have less genetic variability in there. So meiosis provides very important services for both sides of that equation. All right, meiosis. It's a special division that reduces the ploidy number. It's going to happen in 2 stages, Meiosis I and Meiosis II. Key points. Prophase I, we have recombination. That's crossing over that occurs to create new combinations of alleles. During Metaphase I, the homologous chromosomes can independently assort, giving each gamete a different combination. Meiosis II is similar to mitosis, but there's no duplication of the genetic material beforehand, so that's going to reduce the ploidy. And we will see 4 haploid cells produced by meiosis. Sexual reproduction relies on that reduction of ploidy, but also that increased genetic variability. The haploid gametes are then going to fuse together and reform the diploid zygote. All right, we will focus on the sexual reproduction side in the next section.